Okay, we have done for the natural selection. So for today, we're going to look at these artificial selections. So the main difference here is when we talk about the selections here, natural selection basically means that's the environment factor determine which variations they can survive, which variations they can't survive. So that is meant by the natural selection. So it means something like the gods that they choose, okay, which variants can survive better. For example, okay, agouti color codes can survive better, but the white color cannot because they can't have a better camouflage in this case. Okay, so for today's topic, we look at artificial selection. So artificial selections basically means that humans is the one that act as a selection pressure, choose a particular variety or particular variants or particular variations to survive, and some of the variations that cannot survive. So this is fall under artificial selections. So idea here is when you talk about artificial selection, it must involve this very, very important component called selective breeding. Okay. Now, what's meant called selective breeding? Sometimes humans can act as the selections pressure, okay, by accidentally, in this case, hunting. Okay, hunt for a particular variations. Okay, hunt for, oh, this is nicer, hunt for this. But the question here, if they didn't involve in the breeding, then it's not considered as artificial selection. So artificial selections basically must involve breeding program. Means that you choose a particular variant and then breed them. This is means by artificial selections. Okay, so today we're going to look at how these artificial selections have been done onto the animal as well as the crop plants to increase the yield. Okay, so now look at this. Okay, what are the learning outcomes? We're going to focus here. Okay, we're going to describe how selective breeding or artificial selections have been used to improve the milk yield of the daily cattle. Okay, how we can do this. Okay, basically we choose the variants that can produce a high quality of milk or high quantity of the milk. Then we breed them. Okay, and then we're going to outline the following example of crop improvements by selective breeding. So introduction of disease resistance is very important currently. Okay, for the wheat and also the rice, we want them to tolerate to the disease. We don't want them to be knocked out, okay, by the disease. Okay, so introduce. So how we introduce? So this part we're going to learn in detail in gene technology, where we're going to look at how we can introduce a particular gene, okay, to make sure that the plant now they are disease resistant. Okay, also we can do this. We can incorporate mutant allele for the gibberellins. Okay, we have learned about gibberellin, right? Okay, so synthesis into a dwarf variety. So this one again, we're going to look at the gene tech again, how we can do this. Are you clear? So these two, I'm not going to discuss in detail at this topic, but the next topics we're going to look at is how we can introduce the allele or knock out a certain allele in a plant, so that you can see that they can grow better, produce more yield. Okay, let's for example, some crop plants may need 2 years, 3 years to grow, but now they only need 1 year to give us products. Okay, so our focus in this topic will be this one, inbreeding. Okay, and hybridizations. So inbreeding, in this case, we are going to talk about what is the difference between inbreeding versus outbreeding, and what is the benefit of inbreeding, and what are the advantages of the outbreeding. So basically, this will be the first hour of our today lessons. Okay, so first we look at artificial selections. Basically, human tends to be a god. In this case, we see that the human play the role of the god. Okay, and okay, selects a particular variations. Okay, oh, you can survive, you cannot survive. Based on what? Based on the desirable phenotypes. Means that humans want what? Okay, what kind of phenotypes? For example, in cattle, we want them to produce high quality of milk. We want them to produce high quantity, high volumes of the milk. So in this case, those are the desirable phenotypes. Okay, so artificial selection is practiced in the livestock breeding and also the agriculture. We apply it to livestock together with the agriculture. So what we want to have the high quality products from the farm animals as well as the crop plants. Okay, so how we can do this? To achieve the artificial selections, then the breeding procedure or breeding programs either involve inbreeding and outbreeding, but the outbreeding in this case must be a controlled outbreeding. Okay, so we're going to look at what is the difference, what are the differences between the inbreeding and outbreeding, and then you understand why we need to do a control outbreeding. Okay, so the breeding procedure here must involve a selective process. Highlight the word selective process here. Who selects? Human. Okay, humans will do the selection. Humans is a selection pressure here. Choose a particular desired phenotypes and genotype so that their particular phenotypes will be passed on, or genotypes or allele will be passed on to the next generations. Okay, so we inbreeding. What we're gonna do? Okay, what we call inbreeding here. Inbreeding use an individual within one population or one subspecies. Now, this is very important here is if within a same population. So let's say a male and female, we let them cross together within the same population. Then this is inbreeding. Okay, so inbreeding. So the aim of this process is to make sure that if we continue inbreeding, then we're going to make sure that desirable characters of the high quality are propagated from generations to generation. So basically, let's say, for example, okay, you look at the cattle. Okay, so the one that can produce a high milk, then we breed them. Those that cannot produce the high quality of milk or high quantity of the milk, then we actually do not want them to breed. So humans will choose what kinds or who can breed. So when you use the word when they can breed and reproduce, basically means that they're going to pass on that particular allele that confer the desirable phenotypes, so that the next generation will inherit that particular phenotypes. Okay, so inbreeding is to produce a farm animal such as a cattle. So basically, we're looking at the milk productions. Okay, later we're going to look at the process. Okay, so what is the disadvantage? So when inbreeding, okay, inbreeding, most of the time you're going to have what we call the pure breeding. Pure breeding and inbreeding actually promote homozygosity. Now, what's homozygosity? So basically, when you continue to breed and breed and breed over a long period of time, then most likely they will be homozygous. Okay, now for milk production, it may be homozygous dominant. So it means that no problem, they can produce a high quality of milk, but they may have a certain recessive allele together and cause the disease. So this kind of the decline in the variation basically means that by right, the cattle can grow about 10 years to produce a sufficient milk. But the thing here is because of disease, they may grow until 5 years old, 6 years old, or even before they can produce the milk, they already die because of the disease. Why? Because of the homozygosity. When homozygosity means that the recessive allele may be able to be expressed. Sorry, homozygous recessive means that the recessive allele will be expressed and it will cause the problem. Let's say cancer. It will cause a particular disease, or it cannot tolerate to the high temperature. Okay, or they require more nutrients. So in this case, you can see that the milk production will decrease in the population, and this kind of phenomenon we term it as the inbreeding depressions or inbreed depression. So it means that useless. Why? Because you won't be able to gain much, because they won't be able to survive better. So in terms of the outbreeding, okay, outbreeding battle in this case, but outbreeding must be a selective and controlled outbreeding. It cannot be a simply any kind of outbreeding, because outbreeding will promote heterozygosity. So what's the good thing about heterozygosity? Heterozygous always we claim it as a hybrid vigor. Okay, what's mean hybrid wiggle? So it means that when you hybrid both populations together or both variety together, then they will have both variety. Okay, later I will show you what's meant by the outbreeding, but at this moment understand that, okay, they're going to produce hybrid wiggle. So when they produce a hybrid wiggle, what will happen here is they will then to be able to adapt more to the change of environments. They are stronger. They are able to pass on the allele to the next generations. But this outbreeding must be selective and controlled. Okay, so later we're going to look at it, how we can do a selective and controlled crossing among the individual from two different populations, two different populations. So within the same populations, inbreeding. Okay, so between two different populations, then we call it outbreeding. Okay, so hybrid wiggle, the hybrid wiggle will have the desirable characteristics of the high quality compared to the original parent. The original parent may not have both, but hybrid wiggle will have both. Okay, so in terms of crop plants, yes, we always use the outbreeding to actually give us the high quality of seed and also the high yield. Okay, highlight this, it's very important. Okay, the difference between the inbreedings and outbreeding. So let us go through the answer here. So what's the procedure? The procedure here for inbreeding is a technique of mating two genetically closely related parents over many generations. And this is very important within population. Okay, outbreeding. So outbreeding is a breeding method done by the distantly related individuals selected from two populations. Okay, so it means that there is a gene flow. So what's the objective? So you have to know what's the objective. When talking inbreeding, basically we want to maintain the beneficial traits or desirable traits and develop what we call the pure line. For example, certain dog, it's a variety of the dogs. You want to maintain a pure lines, you cannot mix them with other kinds of the variety of the dog. Means you won't be able to produce a pure line. So in order to produce a pure line, you have to make sure that's inbreeding. Make sure that, for example, for racing horses, so we want to have the pure lines. So for pure lines, definitely we need to use the inbreeding. So it means that within the same population, within the same populations. Okay, how about outbreeding? Outbreeding, we want to produce what we call the hybrid wiggle with the vigorous quality. So it means that we want them to have a more quality and produce higher quantity and quality for us. Okay, so what's the genetic nature of the offspring? So tends or more likely to be homozygous because pure line, pure breeding. So means that homozygous. Okay, highlight. It promote homozygous, but outbreeding will promote what we call the heterozygosity and heterozygous. So I will show you in terms of their genetics later, how we get homozygous, how we get the heterozygous in this case. Okay, so in terms of biological fitness, whether they will be able to adapt or not. So inbreeding is more likely to produce biological lower fitness progeny. So that's why certain inbreeding, you can see that they can't survive in the wild. They have to be carefully kept inside a close proximity. Okay, why? Because they cannot compete with others. Okay, outbreeding is more likely, more likely to produce an offspring with the biological fitness. So it means that they are more able, they are able to adapt to the wild environments. Okay, genetic variations in this case, inbreeding reduce. Inbreeding definitely reduces the genetic variations. Why inbreeding reduces the genetic variations? Because of the homozygosity. Okay, homozygosity. So it means that certain allele will be eliminated out, okay, or disappear in that particular populations. Okay, but outbreeding, it will increases the genetic variation because it promote heterozygosity. So both allele must be there. Dominant allele and recessive allele will be always present in the populations. Okay, so expression of deleterious recessive allele means the disease causing allele. So the chances of expression of deleterious recessive allele is very, very high. Okay, because of the pure breeding homozygosity. But in this case, the chances, okay, of expression of deleterious recessive allele is low by using outbreeding because heterozygosity. So dominant allele still can mask the recessive allele. Okay, with the assumptions here, those recessive allele cause the disease, for example, or decrease the survival rate of the animal or plants here. So adaptation to the environment, basically for inbreeding, low potential, right, to adapt to the changing environment. But here, yes, okay, for outbreeding, the offspring or progeny shows higher potential to adapt to the changing environments. Okay. So if you want to equate it with the natural selections, what kind of selections? Basically here we favor a particular extreme end, right? One extreme end, right? We try to imagine between low production and high production, we favor high production. So it means that it's a form of directional selections. Okay, directional selections. So it results in changing the frequency of the allele and also the genotype. So you're going to change the frequency of the allele, right? Because try to imagine the allele that can cause the cattle to produce high quality will continue in that particular populations, but the allele that can't, okay, that cause the cattle that can't produce a high quality milk, then the allele won't be able to pass on to the next generation. So in this case, you can see that it can change the allele and only favorable allele. So allele that show the particular genotypes on particular phenotypes will continue to appeal in that particular population. So it depends what farmer wants. Okay, so artificial selection can produce a new species of plants and animal as well. So this one we're going to learn it, okay, in the last part when we look at the speciation, how the new species can be developed. It will be the next two subtopics. Okay, so this one later we can discuss. Okay, so this is very general, talking about the inbreeding and outbreeding. So let us look at the first part, selective breeding of the dairy cattle. Okay, so it means that in this case, humans many years try to improve. So humans may not know the genetic basis, but what they know, they only look at the phenotype. Oh, this phenotype I want, breed that. Are you clear? Oh, this phenotype I don't want, don't breed that. So this is a very simple idea. We do not need to know the genetics behind the process. We don't need to look at the genetic background, and also we do not need to look at, oh, this genotype I want, or this phenotype genotype I don't want. No need. You just look at how they look like. Okay. So desired features include what? Docility, means that easier to control. We don't want to have a animal that we can't control them. If we can't control them, then how are we going to harvest, right? Okay, and we want them to have fast growth rate, right? So we can shorten, okay, the market durations, right? Okay, when we can market it. So we want to reduce, okay, the use of the resources. If you try to imagine, okay, if I can shorten 10 years, okay, 10 years growing period to 5 years, then I can save half of the resources. Then we can make more money. So this is one of the desired characteristics. High milk yield, produce a very high quantity, in terms of quantity as well as quality. Okay, you can put in another one. Disease tolerant. Disease tolerant basically means that's okay, they not easily get the disease. Okay, or sometimes you can have the climate tolerance. Means that the change of the climates won't affect them much. Okay, so this is what I mean by the desired characteristics. Okay, so how we can do this? Basically, we don't need to observe them only. We just need to observe them. How we observe them? For example, increase in this characteristic have been achieved by selective breeding. Individual that show one or more of these characteristics, we let them breed. Are you clear? So some of these earlier conferring this feature are passed on to individual offspring. So against the offspring will show the desired features for this generation, will choose for the inbreeding. So it means that only allow those that show that, right? The one that show the desired characteristics. Okay. So over many generations, allele conferring desired characteristics increase in the frequency, and some disadvantageous allele are lost entirely. Okay, lost entirely. Okay. So how we can do this? Okay. So first, what we're going to do here is very simple. We take the male. Let's say, for example, in this case, we look at, okay, they can produce high yield. Okay, high milk production. So let's say okay, so we get the male, we get the female. Now, a little bit of problem here when you get the male and female, okay, this is parental generation, because male don't produce a male. So we don't know. In this case, don't know. But female, we choose those that can produce a high milk. So either big A, small a, or big A, big A. It cannot be small a, small a. So if the female show us small a, basically this one will give us what we call the low milk production. So this one, we segregate them out. We remove them. We don't allow them to breed. So that's why we call selective breeding. If the female give us the low production, let's say, for example, they have this, okay, small a, then we don't allow them to reproduce. So if we don't allow them to reproduce, so means that no chance for them to pass on the small a allele to next generations, right? Correct. Right. Okay. But we do not know yet if from this okay, we do not know yet. Okay. So for the male, the bull here, we do not know. Okay. But for female, yes, we know they must have a high milk production. Okay. So if we breed them, most likely we're going to have 3 different genotypes. Big A, small a. Okay. Sorry. I put this in order. Big A, big A, big A, small a, small a, small a. Are you clear? Okay. So now, if this is a female, if the female easier, female, 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 easier. So you look at the female here, then I know that this one, it cannot. This one low, so we don't allow them to reproduce. But this one, okay, they can give us a high milk productions, high milk production. Can I see that? So in this case, I'm going to make them, okay, in this group, let's say I have the male bull. In this population, I have male bulls. Okay, so if let's say this male bull, the male one, the daughter can produce high milk production. So in this case, you can see that the daughter can. So means that this one, most likely they carry the big A allele, most likely, carry a big A allele. Then what we're going to do here is we let them breed. So basically, we inbreed. Inbreed, basically, they can cross it with a father, they can cross it with a sibling. Okay, so within the same population, the same line, can I see that? The same line. Okay, so basically, when talk about inbreed, okay, we breed it within the same population. So it means that it can be within same family. Okay, within same family, or put in other words, between the sibling, or between the parents and also the offspring. Okay, so this inbreed will continue. So in this case, if you continue for many generations, so most likely the genotype will be big A, big A. Can I say that inbreed? So in this form, we say that homozygous. Okay, we promote homozygosity, and we promote homozygosity. So make sure that, okay, the offspring will show that particular desired characteristics. Small a, this allele will decrease in terms of the frequency. So over many generations, you can see that frequency, not frequency, probability of the big A allele increases, probability of small a allele decrease, or maybe become 0. Can I see that? But this is the bright side of the inbreeding. Okay, bright side of the inbreeding. So what is the dark side of the inbreeding? Okay, so for example, because we only concern, we look at it, oh, we want the milk production, high milk production. Okay, but what is the problem here is we don't look at other characteristics. For example, other characteristics, disease tolerant. We ignore in this case, okay? We ignore. So when you ignore, what will happen here is the inbreeding process may result them to produce small b in this case. Because why? Inbreeding. Inbreeding always promotes homozygosity, right? It promotes homozygosity. So the bad thing happened already. So it means that no longer can be disease tolerant. not disease tolerant type already. So means that a mild infection basically mean the immune system down, for example. Okay, they don't have a good immune system. So in this case, you can see that they are not disease tolerant anymore. If they're not disease tolerant anymore, then they will susceptible, they will die young. So means that no point. Can I see that? No point. Why? Because before they can give us the milk, they already die. And this thing will happen, okay, when inbreeding for many generations until you get what we call the pure breeding line, which show the homozygosity. So this phenomena, we term it as the inbreeding depression. Okay, we term it as inbreeding depressions. Then you cannot continue anymore, because if you continue, also useless, no point. You're going to waste your money. Okay, so this is what I say, that's between the two different. Okay, but the problem here is, okay, the bull, you have always to cross-check with the daughter first. If the female offspring show high, okay, productions, then we know that most likely this bull will carry the dominant allele for the milk productions. Okay, so let's look at continue with our notes here. Okay, so selective breedings of the cattle presents the breeder with some problem. For example, if animal, they take larger and take longer time, gestation period also long. So it means that at the end, you may take long period of time to get the pure breeding. Okay, unlike the plants, it may take shorter period of times. Okay, so and also the bull cannot be assessed for the milk productions. Okay, don't say that bull, oh, the bull can produce a high yield, then we cross them. Impossible, because bull won't be able to produce. So what we do, the performance of the bull female's offspring, we're going to check and see whether or not this bull can be used in further crosses. If, for example, majority of the bull female offspring, they can't produce high yield, low quality, then we know that the bull may not carry that particular allele. Then we don't breed them anymore. Okay, so you can see that now these two populations are totally different. This original wild cattle, can you see that original wild cattle? Okay, in the wild, and after many years of the domestications, you can see that we create the different kinds of different variety of the bull, okay, the cattle. So you can see that they have the very large adult, okay, the adult compared to the original one. So because why? We look at this. So those have a small one, we don't allow them to reproduce. Those with a big one, we allow them to reproduce. So continue for many generations. Then you can see that we will be able to get at a particular variety. Okay, but as I say that over a long period of times, what will happen here? Inbreeding depression will take place. Okay, they become disease intolerant. Okay, what they do? They may have a cancer, they may have a certain disease, or they have weak immune system. So therefore, this animal won't be able to grow, or they may not be able to stay long to produce high yield for us. So in this case, we say that inbreeding depression take place. Okay, so these are for the cattle inbreeding. We look at the crop improvement in the wheat and the rice, where we involve the inbreeding and outbreeding together. Okay, again, as I say, that farmer do not need to know the genetic behind how to do this, but what they do, they pick the best plants, okay, keep the seeds, and then we breed them again for next year. So we continue in this case, we're going to get a pure breedings. Okay, so selective breeding continues to be the main methods by which new variety of crop plants are produced. Now this is something that we have to know. Okay, for example, most modern wheats, Triticum aestivum, so this part we're going to learn it again in the speciation. Okay, when we're going to hybridize them together. Okay, so in some cases we have to use a gene technology. We have no choice with gene technology. What we're going to do, we're going to introduce a certain allele so that the plant now become resistant to the fungal disease. Okay, or they are climate resilient, or they have a very effective way to use the nitrogen fertilizer. Okay, so in this case, they will be able to grow well. Then we continue to inbreed them to pass on this particular characteristic to the next generations. No doubt, inbreeding depression will always take place later. Are you clear? Okay, so in this, what we're going to do, first thing, we use a gene technology. Gene tech do what? We use gene tech to introduce the particular allele. We introduce the particular allele into the plant. Once we introduce already, we're going to continue the inbreeding. Now, you don't do outbreeding now. If you do outbreeding, what will happen? If the allele is a recessive allele, it's going to be masked, right? So we will continue to inbreed them so that most of the plants will carry this particular characteristic. So what are the characteristics? Resistant to fungal disease, bacterial disease, okay, climate resilience, nitrogen fertilizer, they use it in a very effective way. So it means that if you use the effective way, they will be able, okay, or farmer will be able to reduce the use of the fertilizer, so save cost. Okay, now sometimes plants, if they grow too tall, the problem. Why too tall? Okay, because if tall stem means that they're difficult to harvest. Okay, heavy rains will knock out, right? Will not flat them, and they produce more straw, which have incurred the cost to dispose it. Okay, when talk about the rice plant, particularly the rice plant. We look at the rice plant. So what we harvest, actually, we harvest the seed here. So remaining one, the stem, we call the remaining the stem, okay, or we call it as a straw. So this stem or the straw, okay, we're going to dispose them. So first, the genetic technology have been used to produce a shorter stem. When a shorter stem, so means that's easier for us to harvest, less susceptible to be not flat by the heavy rains, and they produce less straw, means that we can save money to dispose it. So how we can do this to the wheat and the rice? So we can introduce, we're using gene tech to introduce what we call the dwarf variety. Okay, the dwarf variety. Okay, so again, involved in the DELA proteins that we have learned. So this mutant allele, they carry this the gene code for DELA protein, so to reduce the effect of the gibberellin, right, on growth. How to do this? We know that's what's the function of DELA protein. We learned about this DELA protein. This DELA protein is a repressor protein. So as long as DELA protein is there, then no gene transcription. If no gene transcriptions means that no enzyme. So if no enzyme means that's no growth. So when no growth, in this case, means that the wheat will remain short. So dwarfism. Dwarfism produce. Are you clear? Okay. Or another mutant allele can be introduced so that they don't have a receptor for gibberellin. So if they don't have a receptor of gibberellin, then means that they are not able to break down the DELA protein. So it means that as long as DELA protein is there, DELA protein will go to bind with the transcription factor known as the PIF. Are you clear? So therefore, gene transcription again cannot take place. No enzyme, no protein, therefore no growth. So when no growth, basically the wheat will be dwarf. Okay, so easier for us to harvest, less susceptible, we are not flexed by the heavy rains and winds, as well as less straw. So it means that we do not need to spend more money to dispose the straw. Okay, so you can see that we have the different mutations result in the shortenings of the stem, short stem. So better. So how we do this? We introduce, okay? We introduce what? We introduce the allele code for DELA protein. Or we can actually code allele that code for what? Code for the mutant gibberellin receptor. Okay? Or no receptor. Mutants or no receptor. So this part, please go and check out about the gene control. When talk about the DELA protein, it's very important, guys. Okay. Here. So for the rice also, we are also subject to much selective breeding because we want them to actually resistant to the bacterial blights and also the fungus disease. You can see that the bacterial blights and fungus disease. Okay, so again, we're going to introduce an allele so that they can resistant to the disease. Okay, and also nowadays we want to have the plants that can tolerate the extreme climate change. Right? For example, drought, very dry conditions. Then we hope that our plant or crop plant will still be able to continue to survive. So those, when we introduce, once we have introduced the new allele into the plants already, how to do it, we're going to look at it in the gene tag. So in gene tag, we're going to learn how we can do this. But in this context for today, we just need to know that once we successfully introduce, we have to do inbreeding, because inbreeding can help us to keep that particular desired characteristic. But too long or too many generations, what will happen here is you can see that inbreeding depressions take place again. Okay. So now we're going to look at this one of the example when we perform it onto the maize plant, where we involve the inbreeding and hybridization in the maize, so that will produce what we call the hybrid vigo. Okay, hybrid vigo. Okay, now hybridizations here, you have to be careful. The hybridizations, because we have two forms of hybridization. In today's topics, we are talking about the intra species hybridization. Okay, intra-species hybridization basically means that outbreeding between two populations, but same species. Are you clear? Two populations, same species. Toward the end of this chapter, we're going to look at interspecies hybridization. Interspecies means between two different species. Then the concept totally different. Okay, so we look at the maize. Okay, we look at the maize. So maize were originally grown in this Central and South America, but now it's formed a staple crop in many regions of the world. Okay, maize grow best climates, the long hot summer, provide plenty of time for the crops to ripe. Okay, the crops to ripe. So in order to get a very nice one, we always do inbreeding. So always remember, for farmer, uniformity is very important. Introduce a new word for you guys, uniformity. Now, what's mean uniformity? Uniformity, in this case, we want them to have the same harvesting time. Are you clear? Same harvesting times. We want them to have the same color of the products. Can you see that? Okay. So to achieve the uniformity, inbreeding can help. Why inbreeding can help to achieve the uniformity? Uniform products. So why inbreeding? Because inbreeding promotes homozygosity. Can I see that? Inbreeding can help us to promote homozygosity. Okay, so the plant in each generation become inbreeding promote homozygosity and uniformity. So therefore, over many generations, then inbreeding depression takes place. Can you see that the plants become shorter and shorter, and then they won't be able to survive well. So this form, we call them as inbreeding depressions. Okay. Now, but in order to let them to have what we call the more adaptable, okay, more adaptable to the environment, we can allow them to for outbreeding. But uncontrolled outbreeding will give us heterozygosity, give us a hybrid vigo, increase in variations, but you won't be able to sell it because why? Different harvesting time. This kind of product, you won't have it won't cause the consumer to going to buy, right? Can I see that? So now, our aim here, first, we need to maintain uniformity. We need to maintain uniformity. At the same time, we try to achieve heterozygosity. homozygosity. So what means uniformity? As I said, that same product, same harvesting time. Okay, the duration. You don't want one ripe earlier, one ripe means later, right? And why we want to promote heterozygosity? Because heterozygosity will give us the hybrid vigor. It will give us the hybrid vigor, which will be better adapt to the climate change, or to to adapt to the change of the environmental factor. Okay. So to be able to harvest and sell the crop easily, a farmer needs to make sure that the plants must be uniform. So the challenge here is to achieve both heterozygosity and uniformity. Okay. So how we can achieve this? Let us look at this. Okay. So to achieve uniformity as well as the heterozygosity, then we need to carry out this process in the correct, this process in a correct sequence. So what is the first step? Step number 1, we're going to do inbreeding. Okay, so what we mean here? Now, let's say I am the farmer. Okay, so I do have, I'm farmer. I do have my maize, again, my corn. So what I'm going to do the inbreeding for the first line. What does it mean first line? Line 1. Okay, line 1, I only focus in the high yield. Give us a very, very large coat, very nice yield. Okay, so this kind, if you do it for many, many generations, inbreed, for many, many generations, you will get the homozygosity, big A, big A. So what is big A allele here? Big A means that give us high yield. So it means that small a allele recessive give us low yield. So for line number 1, okay, for line number 1, okay, the first variety, the corns, so high yield because we inbreed for many, many generations, so double big A. But the problem here is because we focus in the high yield, we don't focus other. Let's say, for example, okay, let's say, for example, okay, B allele. We don't care about this B allele. B allele, basically, if the plant carry the big B allele, so means that they are disease tolerant. Are you clear? So if they carry small B allele, it become disease intolerant. Okay, maybe the plants easily will be infected by the bacteria. Okay, so because we only focus at the big A, the gene, it gives us a high yield. So most likely the deleterious allele may be expressed. So in this case, you get small b. Okay, so you can see that inbreeding promotes what we call the homozygosity. Homozygous, keren. But over many, many generations, what will happen to this line 1? This line 1 becomes progressively weaker. We cannot tolerate disease anymore now. Can you see that? So if you continue, then no point. They're going to die soon. Okay? Then what we're going to do here, we produce the line 2. Different variety, but they are still the maize. Okay? They're still the maize. But in this case, we don't care about the high yield or low yield. Okay? But we care whether they can be infected or not. So in this case, we take the disease tolerant. And then we breed them, we inbreed them for many, many generations. Okay? If we inbreed them for many, many generations, so now we know that they will carry big B, big B. But in this case, we don't focus about their product. Whether the product is more or less, I don't care, because this one is my line 2. I focus, I just want to make sure that this is tolerant. But if you continue like this, also useless because they don't give us the yield. So inbreeding depression take place again. Those they don't produce. So in this case, can you see that inbreeding always give us homozygosity? Okay, so now we know both line 1, line 2, both of them experience what we call the inbreeding depressions already. So what we're going to do here now, we are going to do cross breeding or outbreeding. How we do this? So this big A, small b, sorry, produce the gametes big A, small b. Line number 2, they also produce the gametes small a, big B. Now, instead of let them inbreed themselves, what we're going to do here is we're going to outbreeding. But this outbreeding is not we purposely, let them randomly outbreeding, but we select 2 different characteristics and outbreed them. Are you clear? So this outbreeding also named as the intra species hybridization. So you can see that's what the offspring going to have. All offspring will show us with big A, small a, big B, small b. Correct or not? All of them. All offspring. And you realize that outbreeding now provide us what? Heterozygous. Can I say that heterozygous, or we say that heterozygosity, promote heterozygosity. Okay, so what is the characteristic? Heterozygosity better, right? Because why? Now you can see that what is the characteristic? High yield. Big A allele give us high yield. Big B allele give us what? Big B allele give us disease tolerant. Do you realize that? So means that the offspring here will inherit 2 different characteristics from the parent. One is the high yield, another one is disease tolerant. So this form of the offspring, we call them as the hybrid vigor. Can you see that? Hybrid vigor. So we achieved the first one already, heterozygosity, because this hybrid vigor will give us the high yield, at the same time also disease tolerant. But can it achieve the homozygosity? The answer is yes, because all the offspring give us they are genetically identical. Correct? All of them give us high yield, all of them give us a disease tolerant. So it means that all of them will show us the same characteristics. So this one we achieve uniformity. Can you see that? The F1 generation give us the uniformity. So we achieve both uniformity and heterozygosity at the same time, but have to be always careful. This uniformity only lasts. Uniformity and heterozygosity only lasts for 1 generation. Okay, why? Because if you inbreed again, if you inbreed them again, what will happen? Then you won't get everything heterozygosity. Heterozygous. Oops, sorry, let me draw. So if you inbreed them again, what will happen? If you inbreed, then you won't get heterozygosity anymore. Why? You're going to get different phenotypes already because independent assortment take place. And if you don't have this heterozygosity and you have many forms, what will happen here is you won't be able to get uniformity. Are you clear? So if you inbreed these F1 generations, because we do know that big A, big B, small b going to produce 4 different kinds of gametes: big A, big B, big A, small b, small b, small a, big B, and small a, small b. So if you allow inbreeding, you choose 2 and combine, you're going to get high yield, variety of high yield, disease tolerant. This is good. If you get this, it's good. But you may get a high yield, but disease intolerant. Or you may get low yield, okay, disease tolerant. Or even worse, you get low yield and disease intolerant. So overall, you can see that you have 4 different variety already. 4 different variety, so therefore no uniformity. And this one is the homozygous. This one also homozygous, but useless. Are you clear? Okay, can I see that the last one, homozygous, useless. So how to achieve this? So company, we do have this company always help us to carry out the research and sell this kind of seed. Okay, sell the seed to the farmer. So farmer only need to buy the seed and then plant it. They will always get the uniformity as well as the heterozygosity. Okay, so if you need to take a photo, you can take a photo how we can do this. So this is basically the answer for the activities that later I'm going to show you. Okay, so this one means that inbreeding followed by the outbreeding. Inbreeding followed by the outbreeding. Okay, so let's look at the notes here. So we can always cross 2 compatible inbreeds to get the hybrid vigor. So explain how, okay, inbreed and hybridizing in maize promote the heterozygosity and uniformity. Just now I have drawn it already. So those you can copy down. So and explain how we can achieve it. So when one of the maize plant variety, for example, high yield production are inbreeds and repeated for few generations, we're going to get the pure breeding variety of the maize is produced. So the progeny will be genetically identical to each other to the inbreed parent. Okay, so the same process is done to another variety of the maize, for example, tolerant to drought, or just now we say the disease tolerant. So the progeny from the first variety, high yield productions, is hybridized with the progeny from the second variety, tolerant to the drought conditions. Then all progeny are hybrids, heterozygous, but genetically identical. So here I just want to clarify it. So this one is big A, high yield, but not disease tolerant. Okay, or drought tolerance. So this one not so high yield, but disease tolerant. So the offspring will be heterozygous because big A, small a, big B, small b, and all of them, they are genetically identical, so achieve uniformity. But we know that if the hybrid from the 2 pure breedings are inbreeds, the progeny will show no uniformity anymore. Okay, so with this, I have done for the first part, okay, of today's lessons, talking about, okay, I discussed about this selective breeding. Okay, so we achieved the learning outcome here already. We talked about the milk yield production in dairy cattle. We also talked about some gene tags, but I'm not going to detail. We're going to detail how we can do it. We're using gene tags, and we have done the inbreeding and hybridizations to produce vigorous and uniform variety of the maize. And we also talked about how to do inbreeding and also the outbreeding. Okay, so with this, I have done for the first part of today's lesson. I'm going to stop recording.